Thank you. I think the good Lord knew that we would love those big cats so much. Lions and tigers are so gorgeous. And he's like, I'll make them take-home varieties. So I'm pretty certain that's what he did. Who are just as evil as the big ones. They will kill you given the opportunity. They're just not big enough to do it, right? Anyway, I'm going to talk about phylogenetic trees, and so just a few main points here to remind myself not to forget to talk about these things. But we're going to start talking about what a phylogenetic tree is, Darwin's hypothesis versus data. So this is just a little visual representation of what the geologic column is. Now, I mentioned before that basically we have stacks of rocks. So when you hear any of our geologists, Dr. Brand or Dr. Geem, talk about the geologic column, we're basically talking about as you dig down, what do you find? Do you find different types of rocks, and do you find different types of fossils? So here is just kind of a graphical representation, and this, of course, is very simplified, where, you know, at the top we have a specific type of rock, or there might be repeats of this. And then here we have, of course, the millions and millions of years. That's the general assumption whenever we're talking about the geologic column. And the geologic column then is very much used by evolutionists to support their perspective. So as we look at these rock layers, go back here one, it turns out that they're not all just immediately stacked on top of each other, that in reality you can have things that look like this. Dr. Brand showed some pictures of stuff like this before. And this is actually taken from a homework question from like an undergraduate geology class, which is to say which layer came first. And there's some rules of geology that you apply to figure this out. The lower layers came first. That's rule number one. But then when you have things that go through, and then this is the lowest. Did that come first? And it turns out not. This is actually like a magma liquid rock that came up underneath some other time, and then it solidified, and now it's hard rock, but it's granite down there. So in this, a geologist would say, well, B came first, then H, then K, then J, and then there was some sort of shearing event. Well, no, A came next because A is sheared off also, and these things got squished, and then possibly came F, and then came I and D, and then came E, and then came G and C. So it's not just, when you actually look at rocks, this is an actual diagram of what you find in the Grand Canyon in the United States. So there are specific types of rocks. They're given names. In this section, you can see at the bottom of the Grand Canyon, we've got a whole slab of something doing something completely different. This thing is at this angle. Everything else is on the horizontal, and this has got a totally different dip going on here. Something happened that, what looks like happened, is that this stuff came up and pushed this stuff, sometime, and it got bent. So what you actually see in the rocks is much more complicated than this, little stacks on top of each other, okay? So then this, we would say, is a section of the geologic column. This is just a piece of it, and it actually has quite a bit of representation in terms of what the conventional geologist or secular geologist would say is a very long time ago. So Precambrian, those are rocks that really hardly have any fossils in them at all. And then the Cambrian, we start getting fossils, Mississippi and Pennsylvania and Permian. So it's quite a representation of maybe half the geologic column there. So this is more like what you actually see, and this is not even the whole thing. The whole thing keeps going up and on and on. But once again, these are you can break these areas down, and they get more and more complicated. So the Paleozoic is made of the Cambrian, Ordovician, Silurian, and those get broken up into even further breakdowns. So all these, and this is just all available. This is just online. But these areas that we were looking at the Paleozoic, so here's the Cambrian. Look, it's got all these sub-layers. So it's not just that, hey, this is the name of all the rock. No, I mean, there's different areas of the rock that are named different things, and you can see all these are dates. This is a much more technical, much more detailed map compared to what we were showing over here. This just kind of simplified version, and now here's maybe the more technical version that someone studying geology has to memorize. Okay, so good for those geologists, but they had to memorize stuff like that. So you can tell that it's not thrown together. They didn't just make this up. They're out there looking at the rocks. They're out there trying to see what's actually going on there. And then here's the basic. So a phylogenetic tree, and I had to give just a preface about what geologic column is, so that we can talk about why Darwin said what he did, and we can talk about the challenges to what Darwin's proposal was. This is actually possibly the first phylogenetic tree that was ever sketched. This is the only diagram that's in the book on the origin of species by means of natural selection or the preservation of favored races in the struggle for life. This is Darwin's book on the origin of species. And this is the diagram in Darwin's book. And this diagram is basically Darwin's idea in graphical format. Okay? Darwin's idea is that there were down further, there was an original life form. And that original life form then, because of pressures by competition and natural selection and the environment that it lived in, it began to produce, it began to deviate, it began to get mutations, and those mutations were selected for and selected against. So a phylogenetic tree, basically at its essence, this is one from a textbook, is a graphical representation of how an evolutionist would say animals are like each other or related to each other. Now, I'll say honestly, I make and generate phylogenetic trees all the time in my research, and I'm comparing sequences to sequences. And in terms of, remember, biologists, we like to group things and categorize things. We want to know, well, what are these things? Well, and over here, these things are really super simple, so maybe we should group them together. And remember, we were just talking about cats and how they really are all the same. They're just different sizes. They'll all eat you. They should be grouped together. They're similar organisms, right? And so really at the top we have basically just putting things in categories, putting things in baskets. This is a basket of cats. This is a basket of dogs. This is a basket of things with tails, monkeys with tails. This is a basket of primates, they don't have tails. So we kind of put things in categories. And as a biologist, this is taxonomy. This is systematic taxonomy. This is like ground one for what a biologist does, okay? We have to put things together. So the variance here is in making a phylogenetic tree is to say, oh, well, a long time ago, they had a common ancestor. Okay? So a phylogenetic tree almost always then is saying, here was the original common ancestor, and a long time ago those two split off, and this group of organisms became this, and this group of organisms further divided. So this is a modern representation of Darwin's idea. Okay? So this is what a phylogenetic tree is. Now, phylogenetic trees, you can have a couple different types. One type can be based on just sequence comparison, DNA sequence comparison, or protein sequence comparison. This type, it probably has some of that, but it's also got anatomical sequence comparison, which are referred to as cladograms. So when we're talking about looking at fossils in the fossil record, in the geologic column, like all those dead animals, they haven't tried to sequence the DNA because they don't think that there's any DNA in there because they're super old, right? So there's probably not very much DNA there. There may be, we haven't looked, right? But maybe there is. But they had to classify them based on anatomical structure, because they're not living animals, and so we're just trying to classify them. So then phylogenetic trees then are made of dead things, but just on what they look like and what their bones look like, because we don't know much about them. But now we have like little imprints of feathers, and now we're like, oh, these guys have feathers. Maybe they belong in a different group. So the more we discover, the more we're like, oh, maybe we need to reclassify these things. So, and truthfully, I mean, understanding this is pretty important. Protostoma versus deuterostoma. Those are weird, crazy words, but that means as you're developing, you're a ball of cells, and then you become a hole, you make a hole. Does that hole become your mouth or your behind? If you're a protostome, that hole's your mouth. We are deuterostomes. The very first hole that gets made is that end, not this end, okay? It turns out that you can classify organisms wholly based on, does the hole become the mouth or the behind? Who knew, right? Who knew? So it is an interesting reality about how we classify organisms. And of course, radiata versus bilateral. Radiata, I mean, those are 5-sided, sea stars. Okay, you can cut them like a pie, and they're mirror images, symmetrical. There we go. That's the word I'm looking for. Whereas those of us who are bilateral, you can only cut us in one plane. There's only one way. If I cut me this direction, I'm not a mirror image anymore. I can only be cut down this direction, and now I'm a mirror image. And even so, that's not true on the inside, right? So it's only true on the outside and not so true on the inside. And this is another phylogenetic tree of where mammals, and of course, Australia is overrepresented because you have such wild animals here. We're like, what in creation? Praise God for making what in creation animals. Anyway, so those guys get their own whole group because they're so unique. So anyway, we're looking at amniot, those who make eggs, and those things that make eggs are all of these guys, okay? Or have, not make eggs, but have an amnion. And so turtles, tortoras, lizards, snake, crocodiles. And truthfully, the groupings together is helpful. It's helpful to understand if we find a new animal, except for the platypus. What do you say about him, right? Anyway, he does those funny things, and so he gets to be with the mammals. But and then all the placental animals that we're here, this group with cats, in terms of what evolutionist thinks about a human, we're there. So all of this stuff then is evolutionary theory. All of that stuff up there is just grouping together animals with similar sequences and similar characteristics. Okay? So grouping animals together with similar sequences and similar characteristics is helpful for a biologist. When I discover something new, hey, whatever it is that's giving me a cold, we go and sequence it, and we're like, that's new. But it's a subcategory of a respiratory syncytial virus. Well, how do we treat respiratory syncytial virus? Well, it's new, but it's similar enough to this that maybe a physician can treat it. So having these categories and having these sequences is helpful, especially when treating a disease, okay? If we're talking about something that causes infectious disease, which is what I like to talk about. Okay, so Darwin's hope is that the discovery of more and more paleontological data would support his conclusions about this phylogenetic tree. But he did say this: if numerous species have really started into life all at once, the fact would be fatal to the theory of evolution. And the present situation in paleontology, and there's ongoing and abundant discovery, that the actual facts here, and this is a diagram that comes right from Dr. Brand's book, Faith, Reason, and Earth History, is that this is the evolutionary theory at the top. This is what what should be assumed based on that theory, that you have a single or a few organisms that you find in the geologic record, the line being what you actually find, everything above the line being what you find in the geologic record, and then below the line being hypothesized animals, that there's no evidence of them, but hey, they must have had a common ancestor, as opposed to what's found in the geologic column. Okay, so, sorry. This line being the rock record, the animals that we could actually find in the geologic column. Okay, so down at the very, very bottom, where animals start showing up, there's a pile of them. Remember what Darwin said? If numerous species have really started into life all at once, the fact would be fatal to the theory of evolution. And down at the bottom of the geologic column, there are lots and lots and lots and lots of unique things. Okay, so what you should see based on evolution, what we actually find, and all of these P's are representing phyla. So the variance in species is very wide and very well represented. It's not a small number of organisms that show up. It's a large variation. Even way down there, chordates. So chordates are a group of animals that humans are put into because they have a stiff rod, a stiff but flexible rod running down the dorsal part of them. So lots of animals have something stiff running down their back. We have a spinal cord. We're chordates. And they find chordates down there. So they're tiny, they're fish-like organisms, but there they are. The very, very beginning of the fossil record, showing up way, way, way down there in those very, very low rock layers. So to me, this is really, really important piece of data. This is called the Cambrian explosion. Every secular geologist knows about it. Every secular geologist recognizes this is a major problem for evolution because it's difficult to explain. Darwin himself said, yeah, evolution really couldn't be true unless the data supports it. If the data says, look, way down there's only a single organism, and then as you climb up in the rocks, that organism clearly begins to change and it grows limbs. It was a sponge, or it was a sea star, and then it got feet, and you can see that sea star, some with feet and some without feet, and then some with arms. That would be the type of thing that should be expected from the evolutionary theory. That would be the type of data that would be expected from the evolutionary theory. That's not what we find. Instead, we find animals basically fully formed, and they do change as you move up the geologic column. There is some change in them. Most of the time they just stop and another animal starts. And it stops and another animal starts. We don't usually see this gradation of it becoming something else. Really, that's not what we see. So I feel like phylogenetic trees, and compared to the data and compared to the hypothesis of evolution, this is really, really a strong argument that the evolutionary theory cannot account for all the data that's actually out there in the world. Okay, so I'm going to talk just briefly about these guys. These are some organisms called trilobites. Maybe you've heard of them or seen them. They are found very, very low, and for a long time, not a long time, for a very thick layer of rocks. They're not the very, very first organism found, but they're way down there. Okay, and I'm not a paleontologist or a geologist, but if you take a look at, I guess I showed this to show you where they're found. Here we go. So they're not at the very, very bottom of the Cambrian, but nearly. Okay, and they're found for a long time in here. And once again, I'm saying long time because that is the conventional model. They're found for a thick layer, which means that they're found in a lot of rocks. And if you look at their eyes, they're arthropods. They have eyes just like common arthropods, just like the fly. So the two pictures up top, those are fossils. Okay, so we're talking fossils that on the conventional model are something like 200 million years, 300, 400 million years old. Okay, so super old. So, but their eyes seem to be identical to the compound eye of flies. Okay, so what do the compound eyes of flies have in them? Each one has a lens, and attached to each lens is a neuron, a nerve cell. Okay, so flies, their eyes, and responding to the things they see is what takes up most of their brain. Okay, so flies collect lots of visual information, and there's a neuron coming from each one of those lenses, and that's transmitting information to the itsy bitsy fly brain. So here's a picture of a neuron. Here's a picture of the itsy bitsy fly brain. And these are some neurons in insects. So these things have been lit up. And if a fly has those neurons, most likely the trilobite had those neurons. It was a successful animal. It wasn't blind. It had these two. Neurons, from a molecular biology standpoint, are the most complicated cell known in the world. They are a very, very complex cell. They're not single-celled organisms. They have to have whole support systems. They're divas, of the cell world. These guys are divas. They have a whole entourage of people who take care of them, who give them green M&Ms. Whatever it is that they want, neurons get, because they have a group of cells taking care of them. They are highly specialized and obviously very, very important. And in our body, neurons are what make up, that's what's in our brain, right? I mean, those are the cells that carry out the business end of you thinking, is a neuron. And these are the cells in the fly that carry out the business end of them not getting caught by your chopsticks when you try to catch them in chopsticks, right? A fly is like, no, I mean, it has a huge amount of its brain power dedicated to understanding what it sees. So these highly, highly specialized diva cells, really unique, really powerful cells, are found at the very bottom of the geologic column. The stuff down there is not simple. The stuff down there is not early forms of life. The stuff down there has the most complicated form of life that we know of. Way, way down there in the geologic column. So to see this variety, this is not, like I said, it's not even a single cell organism way down there. And there are places where there's single-celled organisms in the geologic column, certainly, and way down there is one. But to recognize that this trilobite, because of that compound eye, clearly has those most complicated cells. And that doesn't follow the evolutionary theory either. These complicated cells should have taken a very long time to show up, and yet here they are down there. So, like I said, I'm not a paleontologist, but just the molecular biology of what these cells are is really, really important. So here is a diagram. Once again, at the line is where we actually have data. So trilobites, and so we're looking at the rocks, basically, in the above rocks. All the stuff down here is an evolutionary hypothesis. So we've never found an organism that's half mollusk and half annelid and half arthropod, or a third. There's no evidence. Yeah, there's no data for that. This is hypothesized. Whereas everything above the line, we do find. We find humans, we find plants, we find sea stars, we find arthropods and annelids. And way down here, we find the trilobites, and those trilobites then have these very, very complicated cells. So that, once again, speaks to that the geologic column is recording death. And it's recording death that took place, basically, from my perspective, during the flood. It's not, however, does not seem to be recording the very simple to very complex evolutionary process. Instead, we find very, very complex and wide diversity way down at the bottom. So once again, I mentioned last talk, the reason that we're telling you these things is so that you can then go back to your students and say, yeah, there's some things that maybe I don't know how to explain. Radiometric dating, we're still like, that's a big one. We Dr. Geem was talking about that, and Dr. Oscarson knows a lot about that. But there's other things that we do know very well, that we know very well support a short age. We know very well support the idea that at the very beginning when animals showed up, they were complicated and they were diverse. It wasn't a single organism that spread into other organisms. So things that you can take home and tell your students, that it turns out that evolution is not supported by all the data that's out there. There is lots of data that is contradictory. And by the way, this, like I said, this Cambrian explosion, the sudden appearance of all these animals, very, very well known, and kind of a thing that secular geologists will actually say that the dirty little secret of paleontology. Those words have been used to describe the Cambrian explosion. Let's just not talk about it. Although obviously they know that it's there. Okay, so I'm going to now talk about a little bit about sequence comparisons, because phylogenetic trees at the one whole group of phylogenetic trees, type of phylogenetic trees I generate all the time, are comparing sequences to sequences. So I want to talk about comparing sequences to sequences. I don't know if you have veggie tails here, but anyway, if it doesn't have a tail, it's not a monkey, it's an ape. Have you ever heard that? Anyway, there's a song, cartoon. If it doesn't have a tail, it's not a monkey, it's an ape. Okay, so imagine that's a sequence. Maybe it's a DNA sequence, maybe it's a protein sequence. Okay, and we're going to compare. If it doesn't have a tail, it's not a monkey, it's an ape. Compare it to the second sentence. If it doesn't have a tail, it's not a story, it's a quote. Okay, that means very different things, right? But it has the same number of words and very close to other, So if we were comparing these two sequences, let's say we have one from a sequence from a chimpanzee, a sequence from a human, you can say, they're similar. There's some differences. But if we compare their meanings or their functions, there might be profound differences. And then let's look at the third sentence. If the prose fails to convey a moral, is it anything more than random words? Now that sequence is very different. You can't compare it one to one. There's a different number of words, a different number of letters. How do you even compare that sequence to the middle sequence or that sequence to the top sequence? Okay, well, they all start with if. So they're somewhat similar. Functionally, the middle, the second, the middle, and the bottom are more alike because they deal with the same subject matter, right? If it doesn't have a tale, it's not a story, it's a quote. If the prose fails to convey a moral, is it anything more than random words? So we're both talking about writing, something that's written, or some communication, versus the top one is not talking about communication at all. It's a way to remember primates from monkeys, right? But it doesn't have a tail, it's not a monkey's ape. So when we're talking about sequence comparisons, this is a very, very excellent example of how difficult it actually is to compare monkeys or apes to humans. It's like, well, is the function the same, or are we just looking at numbers of letters? Are we just looking at numbers of words? So sequence comparison has some nuances. So here's just a little diagram I got from the National Human Genome Research Institute and Science Magazine about comparing 98.8% genetic similarity to chimpanzees, which Dr. Geem has addressed in his workshop. Very excellent addressing of that, which turns out not entirely to be true, and that's what I want to talk just a little bit about right now. The human and chimpanzee Y chromosome sequence comparisons. So if we are comparing these guys to each other, this is a comparison of the human Y chromosome, chromosome 21 and the chimpanzee chromosome 21. So if we just compare them spot for spot, letter for letter, the human chromosome 21 and the chimpanzee chromosome 21 are very similar. Now recognize that chimpanzees have cells very much like we do. They eat food like we do. They metabolize things like we do. And so to keep a cell alive, to keep a cell functional, it has to have a certain number of genes that are just keeping it alive. And I'm talking about eukaryotic cells, so cells that have a nucleus and cells that have membrane-bound organelles. But even cells like bacteria, they have to do the same thing. They have to eat. Okay, bacteria have to eat something. So once you eat something, if you eat sugar, you have to have a way to deal with that sugar. I mean, you don't just put sugar in your cells and burn it. I mean, there's not like little furnaces in there that are burning sugar. You have to have proteins that break it apart and gather the energy from the sugar. So those proteins are essentially identical in bacteria and humans and chimpanzees because we all eat sugar. We shouldn't all eat sugar, by the way. But we all do. We all eat sugar, and the bacteria eat the sugar, and they've got to break it apart and get energy from it. And the human eats the sugar, and it's going to break it apart, make fat out of it. But anyway, it's supposed to make energy out of it. And then the chimpanzee's going to eat the sugar, and it needs to break it apart and get energy for it. That process, just the fact of being alive requires a lot of enzymes and a lot of proteins and to keep that cell alive, you got to have a lot of genetic information to make that cell function, to make that cell be able to eat sugar, to make that cell be able to replicate its DNA so it can divide, to make that cell, whatever that cell is doing, just to make lipids, to make a membrane, to make its physical properties, it has to have a lot of genetic information. And it turns out that the good Lord used those same building blocks in bacteria and humans and chimpanzees and banana trees. If you look back at this, humans and fruit flies have 60% similarity. Humans and banana trees have 60% similarity because we're all made out of cells. Okay, that's really the answer there. In order to keep a cell alive, you have to have what we basically call housekeeping genes, and molecular biologists refer to them as housekeeping genes, genes that keep the cell alive. They build the house. They make the house function. They do the laundry. They bring in the water. They get rid of the waste because your cells do all those things. Everything you do as a whole organism, your cells are doing. They got to eat. They got to get rid of the garbage, and they got to have mechanisms to get rid of the garbage, and they got to communicate with other cells. All these things they do, and so it makes sense then that we have similarities. Okay, so now what I'm going to tell you is was brought to my attention by Dr. Geem. Thank you. Is to point out the reality that the human and chimp in the chromosome 21 are very, very similar. Now we're looking at the human and chimp Y chromosome. Now remember, human and chimp, chimpanzees are supposed to be humans' most close relative. And we are supposed to have divided from them only like 6 million years ago. The truth is that according to this statement, the name of the paper was actually this. Chimpanzee and human Y chromosomes are remarkably divergent in structure and gene content. There is hardly any similarity at all between human and chimpanzee Y chromosome. So you think, well, who cares? If there's no similarity on the Y chromosome, it's not a who cares. There's no possible way that humans and chimpanzees could be related. It's not oh, this is an anomaly. No, it just can't be. Because where did we get that Y chromosome? If it's completely different than chimpanzee, it's not like gorilla, it's not like orangutan, it's not like raccoons, it's not like kangaroo. It's completely different than anything. So although we do share a lot of genetic similarity with chimpanzees because we're both alive, we have opposable thumbs, we have similarities. It's true, we are similar. But the Y chromosome is not similar. So unless that similarity spreads across the entire genome, how could we be related? It's like we're related except in the Y chromosome? Well, where did that come from? I mean, it's an absolute almost full stop. The reason I'm repeating it, and Dr. Geem already presented it, is because it's really important. It's really important. So when you read a biology textbook that's teaching evolutionary biology textbook, and it says humans and chimpanzees are 98, maybe 96, maybe 99% identical, you say, yeah, that makes sense. Except they're not in the Y chromosome. That means they couldn't be related. So the textbook is telling you information, but you're not actually getting the whole story. So the textbook that's presented to young people, and I'm sure repeatedly, it's in your textbooks when we're talking about primates or when you're talking about evolution, it repeatedly says that humans and chimpanzees are related to each other and they're this much related to each other, but they haven't told you the whole story. And this came out in 2010. It's not cutting, it's not brand new. This has been known. So the people who are writing the textbooks are let's just not put that data in. But that data is as important as the other data. It's really super important. So I wanted to make this point again, that this is some things that have been discovered. This was not a creationist group who did this. These were just people sequencing sex chromosomes. But it is super important, and basically it says that what the theory of evolution really, that we came from a similar common ancestor with chimpanzees, is completely cannot have happened if you look at the Y chromosome. That's what this data says. Okay, so indeed at 6 million years of separation, the difference in the Y chromosome content of chimpanzee and human is more comparable than the difference between a chicken and a human. So they're saying that if we looked at the Y chromosome, we look just like we don't look just like a chicken. It's that they're as diverse as a chicken and a human. Remember, chickens are what they think dinosaurs came from. So they're very, very different from us. Anyway, those are some things about phylogenetic trees. I wanted you to understand what a phylogenetic tree is, a comparison, and I wanted you to, because you'll see them in your textbooks, certainly. Have you guys seen them in textbooks? Yeah, so you've seen them. Even in elementary school, they had them in textbooks. And like I said, they're really handy for grouping organisms together. But the actual stuff in the tree itself, the nodes, the separations, those are hypotheses, okay? Those are not supported by sequence data, or those are just somebody's hypothesis. Okay, so just be aware that what is being taught and what's being said sometimes really is a hypothesis. Okay, but it might be presented as fact. All right, I want to thank you so very much for your time, and bless you.